Happy Diwali! I am Sania Kole, and I hope that you guys have a prosperous, happy, healthy, safe year ahead, whatever religion you have or you celebrate. Diwali is the festival of lights, and just wishing you a lot of joy in your years ahead and positivity. So without further ado, I sort of want to share something that is sort of like a Diwali gift, like a treat in a way. It's something that I love to use. It's Metascape. It helps you do gene set enrichment analysis. So if you have a set of genes, what are some of the properties that they share? What are gene lists that they are involved in as well? So how do you figure out that information on what those genes are doing? And Metascape is one of those tools. So whether you are in public health, bioinformatics, clinician, whatever research you're doing, it can be so helpful to figure out what are those genes doing. And I want to show you how you can do that using Metascape. So let's get started. So again, just wishing you a very, very happy Diwali. Very, very happy Diwali to all of you guys. This is my favorite. This is really cute. So yeah, just very happy Diwali. So this is the Metascape. So it's a gene annotation and analysis resource. So again, let's just say you have a bunch of genes and you're trying to figure out what they do, what is their function, what is annotations that they have, what are those? So let's see how we can do that. So I'm going to show you an example where you can use gene names and by themselves, or you can use Entrez IDs and you can query different types of species as well. So let's say that you have a set of genes by their names as well. These are gene names. So there are about, these 34 gene names here. And then we can also eventually look at, what if we look at the Entrez IDs, which are just gene IDs, and I'm a huge fan of those. So if we have this list, let's just, copy this list here. Then we can just paste this list of genes over here. So you definitely need quite a few for it to be meaningful. And so you just paste your gene list here on metascape.org. So metascape.org. So it's a gene annotation and analysis resource. So you just click submit here. And, you don't have to adjust this at all. But, you can look at, the M at musculus sounds muscular. It sounds like a muscular, but it's actually a mouse. C. elegans is like a worm. So there's all these species here. H. sapiens just refers to humans. So then once you put this in here, you do submit, then you can do any species at all, and then you can adjust the species if you're interested in anything. This kind of shows you how many genes were found in that species. So about 32 were found in mice. So we do a lot of studies with mice typically, as well, because there are a lot of similarities. Then this is H. sapiens again, which is humans, and you can do express analysis. So ID analysis, gene enrichment. So this is a tool that I really, really like. It just allows you to figure out from a bunch of different sources, what are those genes involved in? So if you have that list of 35 genes, all these genes here, it's going to query a bunch of databases that people have come up with on different gene functions. And it's, people have functions for neurons dying, for cellular transport, for neuron growth, for lung cancer genes, breast cancer genes like BRCA1, BRCA2, for muscle movement, for Parkinson's disease, and they have from different experiments as well. So it's just taking a while to do some gene enrichment. Okay, it's still going, and then you'll have an analysis report page after that. So what you'll see here is this will give you a Metascape gene analysis report for the gene list that we have. So it sort of tells you that, you put, it also supports Entrez gene IDs or, gene names. And what you'll see here are the results from gene ontology. So that's one of the databases here. So the list of genes that I put in here, these list of genes are involved in like cytokine signaling in immune system, which is huge. Sending the signals to activate immune cells. Leukocyte proliferation, small GTPase mediated signal transduction, pertussis, I'm not sure what that is. Myeloid leukocyte migration, neutrophil degranulation. So like these are definitely leukocytes. These are like immune related genes. And you can kind of maybe see that from the CD genes as well, and IL interleukins, interferons. So those are a lot of different immune genes. Positive regulation of organelle organization. So basically a lot of these genes, they have like these gene lists, they have a bunch of genes that are associated with them. So in fact, if I actually look here at like leukocyte proliferation, so leukocytes are actually, they're actually these white blood cells, these immune cells in our body. So if we just like look these up here, so like that's what a leukocyte is. So if we want to see what leukocyte proliferation, because I mentioned that these are all different gene lists that are queried. So you might be wondering, well, what does a gene list look like? So I can just say leukocyte proliferation. Proliferation is the spread of those white blood cells as well. So we can just click here because it is gene ontology. That's what GEO stands for. There are all these databases and ontologies that are there. So we can just look for that here. And then when we get there, we can click this gene ontology. I know this is for the mouse genome, but I just wanted to show you an example. And what this term has a definition in the gene ontology, GEO. And the 3 branches: molecular function, biological processes, cellular components. So what this is the definition is it's the expansion of a leukocyte population by cell division. So we can also verify that this is 007007, like 0661, and that is the same over here as well. So it's the same term. And again, it's just the expansion of a leukocyte population by cell division. So, the white blood cells are dividing as well. So, and there are all these sub, groups under that, like there are different types like macrophages, monocytes, basophils, neutrophils, and all of those under it as well. So, you can see macrophage, mast cells, leukocyte, macrophage, all of these are under it. But you can see that, and there's subheadings as well, and you can keep going down that too. Microglia are in the brain immune cells. So that are macrophages. And then these are 360 genes that are in this set. So you can have sets, subsets, and any set as well. And this set, or you could even have for any of these, or any of these, So in general, this has 360 genes that are in this gene set, and we were querying with like 34 genes. We were querying again with 34 genes, unique genes. So when we click this list here, we get taken here, and you can see that these are the genes that are 360 genes that are involved in this process. And they have about 560 annotations as well, but you can see the genes that are actually involved. They have 568 and 598, because some of them like BLK, they have multiple enrichments and annotations that they are involved with, but there are 360 genes in this list. So what happens is the 34 genes were significantly about 6 out of our genes in our list were found in this list of leukocyte proliferation. So you can see this is the figure here, and it tends to show there are not as many enrichments for this gene list, but sometimes there are like up to the top 100, and it shows you the enrichments that are the strongest. So you can see that this leukocyte proliferation one we found has 360 genes, of which 6 out of our 34 genes found in leukocyte proliferation. And there are 360 genes in that gene set. So that's like the overlap. And then they do some hypergeometric tests to see, is that significant? So they just run them to all these different lists as well. We can even look at another one like activation of GTPase activity or positive regulation of organelle organization. So we can even look at small GTPase mediated signal transduction and see what does this gene list look like here. So I looked it up here and we can go to the same source. And we can see that any series of molecular signals in which a small monomeric GTPase relays one or more signals, and this 007264 is matching with this here. And how many genes? So 428 genes are in this. And that's this gene list as well. And they can have many annotations because many of the genes can be involved in multiple functions as well. So this one is like AATK, apoptosis associated tyrosine kinase, which is Rab protein signal transduction, happens in neurons of cerebral cortex. So these are all very interesting. This is what we've learned from mouse studies. So you can just see that each of these are different gene lists essentially. And you can click this. You can also download the PDF as well for this enrichment heat map. And then it will just show this to you too, cellular processes, negative regulation. So this sometimes gives you even more because what Metascape is just doing is showing you the top level. Just to make it easier, you can copy, you can save this as, and then you can see that there are a few more like response to stimulus. So we have gene lists for so many things: positive regulation of biological process involved in interspecies interaction between organisms, negative regulation of biological processes, so many different things here. And it goes, so it's updated as of November 1st, which is just a few days ago, given that it's Diwali. So you can see that they look at the gene symbol, short description, they look at biological ontology, UniPro, Protein Atlas. It sounds so much like Hallmark, like the Christmas movies. I'm already thinking about next month. In fact, it's actually Hallmark gene sets are actually found in MSigDB, which is molecular signatures database as well. You can see Hallmark gene lists. You can search for so many things in this, like neuron death, any of the gene lists as well, neuron. As well, apoptosis. And then you can see, this is another thing too. So you can look at Hallmark apoptosis, which is gene-mediated programmed cell death by activation of caspases. So this is one of the gene lists that, and there are thousands of gene lists out there that are being queried to see if they have a statistically significant overlap. Because essentially we have two things that are going on here for each gene list. We have our list of 34 genes here. Like our 34 genes. And then we also have, like, for each gene list, like for instance, this has 161 genes here in this list, 161 genes. And this gene list, okay, for apoptosis, which is cell death. So what we want to see, is there any overlap between these? And is it significant? Like if maybe 3 genes, for instance, are found in, and actually there was no finding here, but let's say that just 3 genes and were found in to map out of these. Is that significant? Like, given the size, so like 3, if there's an overlap of just 3 genes in those lists, is that significant? So there's statistics out there that tries to see, is that a significant overlap? Like, just 3 genes, 3 out of 161 genes, and then 3 out of our 34 are found in that. Is that a significant overlap? That's kind of what those tests are seeing. So 3 out of 34 Is that a high enough proportion of genes in our list? Is that high enough? And with this gene list on apoptosis, on the cell death, 3 out of 161, is that high enough as well? And is it higher than just random chance that you would fall into that list? So that's a kind of test that it's trying to do to see if it's a significant overlap or not. And I can show you that this list, it looks something like this. So genes mediated programming cell death by activation of caspases. So you can see, again, this is another example of gene lists. And you can see here that, the 163, but the first 2 rows were just headings. So there are 161 genes, and you can see, oh, these are like really key genes for Alzheimer's disease as well, but they have a lot of different functions. So, and these are caspases, which is what it's sort of talking about, caspases. So that's just showing you a gene list again, and they query all these lists, and you can also just take this here. And these values here are kind of what is shown in these bar plots as well. They're plotting this negative log 10 p-value. So for each given gene list, pathway and process enrichment analysis has been carried out with the following ontology sources: KEGG pathway, gene ontology biological processes, reactome gene sets, canonical pathways, quorum, TRUST, which is transcription factors, this GeneNet, disease gene database, PaGeneNet, like patterns in tissues, transcription factor targets, wiki pathways, and even COVID. So based on, if you have a gene list of genes that you've run an experiment and they are found significant in COVID pathways, then, and you can test them across all these data sources. So it's not just this one, it's not just this list, it's not just this list, it's so many lists that Metascape is kind of testing them against all of these were found as an overlap between, cytokine signaling in immune system and R34. So that's pretty high. So it says all genes in the genomes have been used as an enrichment background. So it looks at all genes that we, it's saying by random chance, if I just selected 34 genes from the genome, from all of the possible genes that they are available, I think at least, 20,000. If they randomly selected 34, how would that compare to how likely are they to fall in each of those gene lists, and how many actually fell in those gene lists? So for instance, we have a set of our list of, again, we have our list of 34. And then we're gonna have random 34 that are selected. And what it's trying to do is it's trying to, and then we have like, I just put it here. We have this like gene list. Biological gene list. This can be anything. Like for example, cytokine signaling in immune system. So out of this list of genes, or however many genes it has, we're finding is that there are 8 that were found in it. There are 8 of our genes, 8 of our genes that are actually mapped between this. 8. That's what this is saying. 8. And that is significant relative to like a random 34 genes from the genome. That is even more like having 8 from 34 fall in this list is relatively high than looking at over 20,000 genes, randomly selecting them, 34 genes, and you generate some random list of genes. The odds of them overlapping is kind of slim, but you generate 34 random genes from the genome, and then you have your list of 34. And then you just see if there are 8 here, and if that is high enough overlap as well, 8 out of the 34, and then 8 out of these many relative to what you get randomly. So terms with a p-value less than 0.01, a minimum count of 3, so at least 3 genes need to fall in from the gene list, no matter what the size is. It needs to be at least 3 genes from your list need to fall into that list. An enrichment factor greater than 1.5. So this is an enrichment factor, I think. Is a ratio between the observed count and the count expected by chance. This is the count expected by chance is from the random. And observed count is given by this, 8 here. And maybe by random chance, we might expect maybe just 1 to fall into that list. Maybe by random chance, we expect maybe like just 1 falls into that list. So that's what it's sort of comparing it against. Maybe by random chance, we just get 1. But because of, that this actually has a lot of immune genes, 8 were found in here. And that is so much higher relative to this. So the ratio is 8 to 1. So it's 8, for instance. So the enrichment factor is greater than 1.5. So we include it. There are at least 3 genes there, and are collected and grouped into clusters based on the membership similarities, p-values using the accumulative hypergeometric distribution, q-values using the Benjamini-Hochberg procedure, multiple testing, because we're running multiple testing across multiple gene lists. So you're trying to control for the fact that you could just randomly have so many false hits, like, enrichments that may not be meaningful, but just by chance. So you do some testing correction as well. And then we do hierarchical clustering. So basically, all you should know is that we're just seeing our gene list and every gene list. We're querying not just cytokine signaling. There was 8 here. Then leukocyte was 6 out of those 360 were found as well. So we're just trying to see if that is significant relative to the size of this, relative to this size, relative to what we can get randomly as well. So then these are just outputted in this enrichment figure, so you can save it. Then you can also see that they have, they are also doing Cytoscape visualization network of enriched terms as well. So you can see that this is how these terms are connected to each other in a network. Then there's protein-protein interaction enrichment using string. It's a database of protein-protein interactions. So we can see that these are just genes and how they're connected, by cytokine signaling in immune system, hematopoietic cell lineage, KO, KEG ontology. Then this is HSA, which is, I think some other pathway. And that these are what these are involved. Of enrichment analysis in COVID. And what we can see, I looked at the paper for this too and talked to the author, which is cool. Like they're like sometimes celebrities in my mind because they do such cool work. So talking to them, these academic big shots is often really nice and cool. So this is COVID-17, and you can see that 3 out of these genes in our 34 are found here. So, and this is a study, and they identify genes for COVID that probably go down in the lungs. And that's a study. Maybe one day they can be like a quitter. So so this is the RNA Blanco Mello lung down. Yeah, these are all these COVID lists that are out there. This is DisGeneNet, which is like a disease gene network database. And they have and it just basically queries all the diseases. People who organize that database basically are focused on diseases, and you're trying to see, are those list of genes involved or associated with those diseases? So, this is some myelodysplastic. So they all go from, the significance down. And even if there are more counts that are found, you might be wondering, well, why doesn't this 8 go above that one? The reason is that what if like your gene list is smaller? So we have our 34 here. So we have our 34. And what if like we're comparing it to a gene list that is kind of small, like one gene list may just have, like, let's say that this is biology list one has just like 20 genes in that list. And the other list could have maybe way more. This list could probably have like 570 genes in this list. So this is like some biology list too, maybe. It could be some disease, whatever it is. So even if you have more overlap between this list and this list, there are so many more genes, so you're still likelier to get more overlap between these two. But that doesn't. But what if like 5 out of these genes are found in this? Like what if, for instance, like 5 are found here? Oh wait, let me just make the colors better. What if 5 are found here? And then what if, and that's what statistics kind of test for. Is it really significant, you know? And what if like just 10 are found here? Maybe 8, hypothetically 8. So even though this number is higher than this, it's that there is 5 out of 20. So about like 25% of these genes are found in these. Whereas 8 out of 570, so much smaller proportion of these genes are found in this. So that's kind of also what goes into it, like relative to the size, like how many genes are in there? Relative to this size, how many genes are common? It's just like a Venn, it's just basically, it's a Venn diagram analysis here too. You have like your list and you have another list, and you're also seeing, so this is like, you know, your list. And this is a bio list here. And you're focused on this part here. But you're also kind of comparing it, the same list, you're also looking at it from, like you're also seeing if you didn't have this your list, how much would you, what's the commonality between them? But if you also randomly came up with a list of the same size from just randomly looking at the genome. So if you had a list from this of the same size, so like if we have 34 genes, so this is, you know, this is also random 34. And then this is your list of 34 genes. And this is random 34, and then that's the same bio list here. How do those compare, basically? You just kind of want to see, like, is it by random chance, do you have fewer? And it could maybe the random chance that you maybe have 0 and random the same size. And you're kind of also comparing these two to each other. You're kind of comparing these values to see like the overlap between those two lists of genes. Wow, it looks like a complicated web. So while we're at it, I'm also going to draw my favorite logo. Yay! Yeah, that's actually my favorite logo, the little star. Okay, so we're going to go back to this. And you can see that all these diseases like allergic conjunctivitis, as well, Hashimoto disease, tumor immunity, allergic asthma. So then you also have this list, and basically it's plotting these negative log 10 p-values here. Then PAD gene database is also looking, you know, tissue specific. So we can also just see what does this do. This PAD gene database also just goes here. A pattern gene database, you know, looks on genes under various tissue and time specific conditions. As well, integrates pattern genes quantitatively identified under various tissue and time specific conditions. So it's just focusing on that. And then you can also see this as well, which is, you know, it's finding 13. That's actually pretty high. So 13 of those genes are found in the spleen. Yeeks, my spleen. No, and then also in the blood as well, there's 6. And then these are transcription factor targets. So what this database looks at is looks at transcription factor targets for those genes. So from transcription factor binding site profiles as well. Low throughput transcription factor functional studies, like targets, how transcription factors interact in a transcriptional network. So it's just looking at those targets here and trying to look that, oh, many of these genes, 5 of these genes tend to be targets of MAML1, which is a transcription factor as well. It's a transcriptional co-activator. And we can, so MAML1, and 5 of those happen to be target genes. So you can see all the sources that they're looking at here. And then what you can also do is you can also just download the Excel. You can download a gene list report PowerPoint file. I also want to show you something else. You can kind of see that this is the analysis report part, which you can download. You can download the Excel sheets with more information and a zip file and a session ID that can last for 72 hours in case you want to come back and don't want to wait for the enrichment to run again. Then you can see a gene list analysis report page here. Each slide has notes that explain the analysis, which basically tries to take whatever's in Metascape for the most part and put it in a set of slides. Like your gene list has 34 identifiers, 34 human Entrez gene IDs. These are, it tells you how it got all of these, gene enrichments, ontology clusters, what each thing represents. It's kind of cute. It provides these for you as well. Each M code network is assigned a unique color. What are these annotations as well that, you get from here? What do they mean? And yeah, just a bunch of different analysis you can get too. So that's also really, really, really cool, I think, the kind of analysis. So I'm going to show you another example of just how instead you can do this type of analysis if you have just Entrez IDs for information. If you can also run these same results just for the Entrez IDs. So basically, if we go here, you can see that these are Entrez IDs for the same set of genes here, the same set of 34 genes. We can instead also use these Entrez IDs, and sometimes those can be even more robust. So we're going to go back to Metascape, and then I'm going to show you how, oh gosh. So we're going to go back to Metascape, and I'm going to show you how we can kind of just, put these in a list using the Entrez IDs as well. So we can just put in this list of IDs as well, and that would work. And it detects 34 Entrez IDs. These are gene IDs, and I have videos where I talk about how you can get those IDs and vice versa, go from IDs to gene names potentially. So yeah, you can see that. So it's basically a biologist-oriented resource for the analysis of systems-level data sets. If you have a lot of gene lists, you want to figure out what they're doing. And you can see the improvements since I started using them. String, eggnog. Oh, that sounds so festive. In the meantime, yeah, you can just save them, download them. You can load them up in Cytoscape or on a web to play around with networks that you get. Acute leukemia. You can see all these different databases here, and we're just still waiting for, okay, now it's finished running these, and you'll see that it hopefully should be the same set of results as well. So it's the same cytokine signaling, all this good stuff here. And then you can also see the Excel sheet. So let's check out what we find in Excel. So we get this Metascape results in Excel that are also great. So that's great. So it tells you, okay, this is your list, this is your gene ID, Homo sapiens, so, and it actually is cool because it also tells you what the gene symbol is. So we gave in, this tells you what you put in. So if you put in gene names, it would have the gene names. And you can put a mix of them in too. Mix of IDs, mix of names, but I don't know why you might do that, but it's all good. And then it also maps the gene symbol for you with the Entrez gene ID. So like, this Audrey gene, it has this ID for enable editing. But for instance, so if we look up this gene here, or add GRE1, brings back grad school memories, you can see that the ID is 2015. So that just kind of so that just kind of shows that the gene ID and the gene symbol, it tells you that information. It gives you a little bit description of the name of the full length name, adhesion G protein coupled receptor E1. So that's sort of where that name comes from. What are the processes that it has been involved with, separated by semicolon. So you can use this information as well, kinase class. And then these are the bunch of the responses that were found as being significant. So these were the ones that we found, like cytokine signaling in immune system, leukocyte proliferation, small GTPase mediated signaling, transduction. I think they kind of truncate the names. These are all of the ones that are significant, and it's sort of telling you that, hey, there were 8 that were significant, which of those they were that were involved in this process. So that way you can also just check, okay, there were 8 here. If you, okay, I think maybe we have to convert these to numbers. Okay, because these are general types. So let's convert these to numbers. Okay, anyways, let's count them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 for this one. Okay, so I think there's a little bit higher that you can also see for each of these. You can get the category, the terms, the descriptions, those log values. So it still says that there are 8 here in this set. And it tells you the 8. Actually, this is better because it tells you exactly the 8 to look for. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 11. So leukocyte proliferation. This tells you the overlap that we have as well. And I think maybe it's weighting the counts. So you can see here that, these are some, it's doing some other complex work, but I'll try to explain the general concepts of how they do these enrichments. And remember, we were looking at the numbers in mouse, but we can also look at it in humans. And I think that this is the term list. So these are just like Excel documents that you can use to also analyze the results that you find from Metascape. So hopefully you can see that you can download the results in the file. You can play with them in Metascape and Cytoscape as well, and in PowerPoint. And I really hope that this is helpful because you can do gene set enrichment analysis. You have a lot of different bar plots that you can use to figure out the functionality of your list of genes. So I hope that this was helpful. And if you have any questions, please let me know. I am Sanya Koller, and whatever you guys celebrate, I just want to wish you a very, very happy Diwali. Thank you, guys. And lots of love and best wishes today and always to you guys. So hopefully this will help you on your research. And again, with any questions, please, please, please let me know. And hopefully the Diwali of Light, this will give you guys a light bulb of ideas of how you can use this to better understand your list of genes, what are gene enrichments, how to figure out what genes do. And in the spirit of a happy Diwali, I just wanted to give you guys some wishes from my little home temple, Father Shivji and son Ganeshji. And I just wanted to spread a lot of positive wishes to you guys this Diwali. In particular, Yangshi also wants to say a very, very happy Diwali as well. Happy Diwali! And wow! So whatever religion you celebrate, Diwali is all about light and happiness and basically prosperity and just being good and letting a lot of light shine into your lives and bring a lot of brightness. So based in that vein, I just want you guys to also have a very successful and prosperous year ahead. And I want to do my part to help you guys throughout as I get help on my journey. So, oh, happy, happy, happy Diwali. Happy, happy Diwali to you guys. So Yangshi is wishing you well too.
Hi, little guy. Say happy Diwali. Happy Diwali. You're the cutest cat. Happy Diwali to you guys.